Hello, my name is Yan Li, and I'm from the Center for Reproductive Medicine at Shandong University. And today, I'm gonna give you a talk about early placental development. So, a brief outline will cover what is the placenta, the early development of placenta, and then talk about clinical diseases related to inadequate placentation. We'll also cover in vitro models to investigate placental trophoblast differentiation. The placenta supports the fetus while it's growing. Firstly, let's take a look at the placenta. We all know that the baby comes along with the umbilical cord, and down that cord, you have two umbilical arteries, which carry the oxygenated blood away from the baby. And then you have one umbilical vein that carries all the good oxygenated blood full of nutrients flowing away from the placenta and into the baby. Inside the placenta, you have complex artwork of arteries and veins. First of all, you have the maternal vein and artery inside the placenta. They feed into something called intervillous space, which is basically pools of maternal blood formed just like a lake where maternal blood converges and sits there waiting to interact with the fetal blood. Then you have the umbilical veins and arteries that form a tree-like structure and penetrate into intervillous space. But inside these pools, maternal blood and fetal blood don't actually mix. They come in very close contact across a thin membrane, and that's known as the placental membrane. So through this placental membrane, lots of things can transfer or diffuse from maternal blood to fetal blood, and vice versa. And it is the process of diffusion that forms most of the functions of the placenta. Development of the placenta begins during implantation when the blastocyst adheres to the endometrial epithelium. Trophoblast cells differentiate into an inner cytotrophoblast layer and an outer syncytial trophoblast layer. As the cytotrophoblasts proliferate, newly formed cells migrate into syncytial trophoblasts and lose their cell membranes, forming a rapidly growing multinucleated mass. The cytotrophoblast secretes proteolytic enzymes, and the syncytial trophoblast sends out finger-like projections, allowing the blastocyst to embed into the endometrium. As the syncytial trophoblast erode the endometrial blood vessels and glands, lacunae or spaces begin to form. The lacunae become filled with maternal blood and glandular secretions, and isolated lacunae fused into a network where maternal blood flows, thus establishing early uteroplacental circulation. Towards the end of the second week, small projections of the syncytial trophoblast, CTB for short, begin to expand into the syncytial trophoblast, STB for short, to form primary chorionic villi. Early in the third week, actual embryonic mesoderm grows into this villi as a core of loose connective tissue, at which point the structures over there are secondary chorionic villi. By the end of the third week, embryonic blood vessels have begun to form in the actual embryonic mesoderm of the secondary chorionic villi, transforming them into tertiary chorionic villi. In brief, the placenta develops from the layer of cells on the outer part of the embryo called trophoblasts. These cells on the outside are able to differentiate into both multinucleated syncytial trophoblasts that secrete HCG and into invasive extravillous trophoblasts, which express HLA-G on their surface, and finally form the placenta that we see at term. Furthermore, the inadequate development of placenta, or more specifically, the dysregulation of trophoblast proliferation and differentiation, is related to many pregnancy complications, including miscarriage, preeclampsia, intrauterine growth restriction, choriocarcinoma, etc. Take preeclampsia, for example. It is characterized by de novo development of concurrent high blood pressure and protein urine after 20 weeks of gestation. It happens in 5 to 8% of pregnancies worldwide. Other than maternal problems, there are also neonatal complications, the most common of which is intrauterine growth restriction. And there is no cure for preeclampsia except delivery. It has been a leading cause of preterm deliveries, maternal and perinatal mortality and morbidity worldwide. Based on previous publications, it is thought that preeclampsia is a disease of abnormal trophoblast differentiation, and in particular that extravenous trophoblasts don't differentiate very well in this disease. The shallow trophoblast invasion is responsible for subsequent improper maternal spiral artery remodeling and poor placentation, leading to the release of soluble unfavorable factors in the maternal circulation that result in systematic endothelial dysfunction and clinical manifestations of preeclampsia. To elucidate the etiology of preeclampsia and look for small molecules or drugs that can enhance differentiation of cytotrophoblasts into extravenous trophoblasts, appropriate study models are needed. It is worth to mention that the mouse has been used very commonly as an animal model to study the development of numerous organs and systems. However, compared to other organs, the placenta is evolutionarily divergent. So it means that compared to human, the mouse placenta has quite different structure and cell types, and in particular, it is quite different in terms of early developmental pathways that regulate its growth. In addition, placental diseases like preeclampsia are actually specific to higher primates. Therefore, the mouse is an suboptimal model for studying human placental development. So, for this reason, a lot of scientists have turned to tissue culture using cell lines in the dish in order to understand human placental development. It has been summarized that over the past 5 years, the most prevalently used cell models for investigating early placental development are HTRA and BU cell lines. Of note, the choriocarcinoma cell lines, including BU, JAK3, and JAR, are highly malignant, contain abnormal numbers of chromosomes. HTRA cells are widely used for studying the invasion and proliferation behaviors of trophoblasts, while BU cells are usually used for investigating the differentiation of cytotrophoblasts to syncytial trophoblasts, since only BU cell line could be induced to secrete beta-HCG. Primary human trophoblast cells isolated from the first trimester of placenta and villus explants are also utilized for functional and mechanist investigations. More recently, human trophoblast stem cells have been derived. They can be differentiated into both syncytial trophoblasts and extravenous trophoblasts. Long-term expanding organoid cultures from purified first trimester cytotrophoblasts or human trophoblast stem cells, also known as trophoblast organoids, are also being demonstrated. In the future, the application of organoid culture system, as well as co-culture of trophoblast with decidual components, could better recapitulate the actual microenvironment of maternal-fetal interface. And every few weeks, many new methods coming out in omics area. These achievements provide us a powerful new set of tools. Also, the interdisciplinary collaboration, for example, advancements in microfluidics to analyze the dynamics of trophoblast migration is developing rapidly. All of these achievements will help deepen our knowledge regarding normal human early embryo development and pregnancy complications such as preeclampsia. So it is an exciting field to be in. Again, thank you for watching today, and I hope you enjoyed this video.